So welcome back to class. We just finished talking about G protein coupled receptors, which are large receptors that are coupled to G proteins. And there's another class of receptor we're going to talk about, our enzyme coupled receptors. These are receptors that have the enzymatic activity built right in. So here you can see this receptor, when it binds to its ligand, right on this enzyme is an active domain on it. So when the ligand binds, it stimulates their kinase activities. And that receptor kinase signal is going to go down through a phosphorylation cascade to other proteins. Mainly, we're going to talk about receptor tyrosine kinases, but there are also serine threonine kinases that work pretty similarly. Okay, so one type of receptor that is a receptor tyrosine kinase is going to be receptors for growth factors. And so for a cell to be able to grow, not only does it need enough nutrients, it also needs to have plenty of growth factors available, which stimulates that signal that it is time for that cell to grow. One way that we get growth factors is from blood clotting. Platelets, when they're activated, will secrete growth factors. Blood will clot in response to an injury, so your body needs to grow new cells to heal that injury. So the growth factors that platelets secrete, which is called platelet-derived growth factor, is going to stimulate fibroblasts to form new connective tissue. And that platelet-derived growth factor is going to bind to a receptor that is a receptor tyrosine kinase. There are several growth factors that stimulate receptor tyrosine kinases, such as insulin, insulin-like growth factor 1, fibroblast growth factor, epidermal growth factor, and nerve growth factor. On this table, you can see several different types of growth factors, and a lot of them are binding to tyrosine kinase receptors. But there's also some other types of receptors that growth factors can bind to, such as this acetylcholine receptor or a 7-pass protein. Many receptor tyrosine kinases are going to lead eventually to the cell growing, proliferating, or specializing. You may remember talking from our cell cycle chapter that when the growth factor binds to the kinase, it leads to transcription of genes that are involved in taking that cell through the cell cycle. And you can see that your receptor tyrosine kinase just has one transmembrane segment. That's very different from the GPCRs that have 7 transmembrane segments. And on the extracellular side of this receptor is a ligand binding site, which binds to the growth factor or whatever its ligand is. And on the cytosolic side, you have a tyrosine kinase, which can phosphorylate tyrosines, and then a lot of tyrosines, which can be phosphorylated. And here, when the ligand binds, you can see the receptors group together and they'll phosphorylate each other. So this tyrosine kinase will make sure all these phosphates are added to these tyrosines, and this tyrosine kinase will make sure that phosphates are added to these tyrosines. Sometimes on our receptor tyrosine kinases, the receptor part and the tyrosine kinase part can be separated from each other into 2 separate proteins. And in that case, we would call it a non-receptor tyrosine kinase. It's still a tyrosine kinase, but it's not bound to the receptor anymore. So that's why it's called a non-receptor tyrosine kinase. But a non-receptor tyrosine kinase is still able to bind to the receptor and be activated when the ligand binds. When the ligand binds, that starts a whole sequence of signal transduction. The first thing that happens is that your receptors are going to come together. For things like fibroblast growth factor, the receptors are just going to dimerize. You'll have 2 of them together and cross-phosphorylate each other. Sometimes you could have a clustering of multiple receptors, such as with epidermal growth factor. But because they are all receptor tyrosine kinases phosphorylating other receptor tyrosine kinases, it's called autophosphorylation. The kinases are phosphorylating the same type of receptor as themselves. Then once autophosphorylation has happened, then we start getting cytosolic proteins recruited. And these cytosolic proteins are going to have to be able to recognize a phosphorylated tyrosine And these domains are called SH2 domains. SH2 domains can recognize phosphotyrosines. And when the SH2 domain, the protein with an SH2 domain comes, it'll be activated and can activate other signal transduction pathways. It can even activate multiple pathways at the same time. You may remember the RAS pathway we talked about earlier this semester. RAS is a protein that's important in regulating the proliferation of cells. When it's active, it will lead to a signaling cascade that activates things like cyclins and CDKs that help the cell get through the cell process. RAS is a G protein. You can see here it can bind to GDP and GTP, but it's not the same as the trimeric G proteins we were talking about earlier. This is the small monomeric G protein. And RAS, although it can bind to GDP, this is its inactive state. For it to be active, it has to bind to GTP. And for GDP to come off and GTP to be put on, it's going to take a guanine nucleotide exchange factor. And that exchange factor is called SOS, S-O-S. So here you can see SOS takes off the GDP and adds the GTP onto RAS. So now RAS is active and can go through this whole signaling cascade. SOS is going to know to replace the GDP with GTP because it interacts with the receptor tyrosine kinase through this GRAB2 protein. So receptor binds to ligand, autophosphorylates, GRAB2 associates with that autophosphorylated region and triggers SOS to add a GTP to RAS. RAS will then signal through a protein called RAF. RAF is able to detach from the membrane, whereas RAS is associated here. And it starts this protein cascade beginning with MEK. MEK is a protein kinase. It'll be phosphorylated. Then it phosphorylates MAP kinase into phosphorylated MAP kinase, which then can activate ETS and JUN, which lead to some other proteins being created. So these are sometimes called, a mitogen is what causes a cell to go through mitosis. So that's where this name MAP kinase came from. It's mitogen activated protein kinases. This MAP kinase, when it's phosphorylated, it triggers these transcription factors, ETS and JUN. They're going to be phosphorylated and activated to go turn on the transcription of genes that are needed for the cell to grow and divide. Then once RAS is activated, it has to also be able to be inactivated. So you're not continually causing that cell to divide. And that is going to be caused by a GAP protein, GTPase activated protein. The GAP protein is going to cause the GTP on the RAS to be cleaved to GDP. And so now RAS is back to being bound to GDP and it'll be inactive again. We can study a lot of these receptors by making mutant receptors and seeing what happens to that cell. Mutations can be caused in the receptors themselves or in downstream signaling components. So, for example, normal fibroblast growth factors can undergo autophosphorylation when ligand binds, but mutant fibroblast growth factor receptors, they'll be able to bind ligand but not be able to go through autophosphorylation. So the type of mutant receptor will be able to interact both with normal receptors and with other mutant receptors and prevent signaling from happening downstream. So here is your normal receptor. Your tyrosine kinase binds to the growth factor, dimerizes and autophosphorylates it. If you've made a mutant, the two mutants can still bind to the growth factor and dimerize, but there's no cross-phosphorylation. What's really cool, though, is that the mutant growth factor receptor can dimerize with a normal receptor, and that is going to block this normal receptor from doing any phosphorylation itself also. So this is a dominant negative mutation. The mutant is going to be dominant over the normal phenotype because the mutant receptor blocks also the normal receptors. Some mutations are going to make them active, constitutively active. So these are constitutively active mutations. These receptors will be always on, always sending the signal, even when ligand is not bound. In humans, there is a dominant mutation for a fibroblast growth factor 3 that causes a form of dwarfism known as achondroplasia. That causes signaling to cause the premature ossification of collagen in your bone. Receptor tyrosine kinases can also activate phospholipase C. Although this is a slightly different version than the phospholipase C we saw earlier. This is phospholipase C gamma. This will still lead to IP3 and DAG being created. In this case, the phospholipase C gamma still binds to that SH2 domain on the receptor tyrosine kinase and has a SH2 domain that binds to the receptor tyrosine kinase. And the receptor tyrosine kinases can also activate PI3 kinases. Okay, I also said there were another type of receptor kinases, the receptor serine threonine kinases. A major class of them is the TGF-beta growth factors, or transforming growth factor beta. You may remember talking about them when we talked about our cell cycle. TGF-beta signaling begins when the growth factor binds to the transmembrane receptor. And you can see there's two different kinds here. There's these, which are type 1, and there's these, which are type 2. But they're all part of this serine threonine kinase. When the ligand binds, TGF-beta is present. It causes these four to group together, and these type 2 receptors are going to phosphorylate the type 1 receptors. So now you have phosphorylated type 1 receptors that are active. And then these phosphorylated receptors are going to interact with SMAD proteins, and they're going to phosphorylate these SMAD proteins. Here we have our SMADs that are interacting with the TGF-beta, and they're also going to interact with SMAD4. So these SMAD proteins, our SMADs are getting phosphorylated from our TGF-beta receptor. So now we have this receptor, or this complex of SMAD4s and phosphorylated R SMADs. This whole group goes into the nucleus and can act to turn on or off specific genes. There are also other SMADs that can act to inhibit the TGF-beta pathway. There's a couple of other types of enzyme-coupled receptor families that we don't know quite so much about. They're a tyrosine phosphatase receptor that remove phosphates. They're phosphatases. They remove phosphates from tyrosine receptors. So that would indicate they're able to regulate our tyrosine receptor kinases. And there are guanylyl cyclase receptors. These act similar to adenylyl cyclase, except they're going to make cyclic GMP instead of cyclic AMP. Guanylyl cyclase receptors are going to be important in the photoreceptor cells of the retina, in fluid regulation in the gut, and in vasodilation. Okay, so there's some more types of receptors and some more signaling pathways that we've talked about. We've got one more video for this chapter. We're just going to kind of wrap it up, integrate the whole thing together. So I'll see you soon. Bye.